Afternoon, everyone. I'm Colin O'Connor, custom content editor at Genome Web, and I'll be your moderator today. The title of today's webinar is Miniaturizing and Automating SARS-CoV-2 Sequencing in an Epidemiological Lab, and it's sponsored by SPT LabTech. Our speakers are Richard Chalvignac, project engineer at Civil Hospital of Lyon, Quentin Semanas, virology research engineer at Civil Hospital of Lyon, and Kamila Koprowska, senior business development scientist at SPT LabTech. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Richard Chalvignac.
Thank you, Colin. Hi, everyone. Thanks for introducing us. So, as Colin said, we are going to make a presentation on miniaturizing and automating SARS-CoV-2 sequencing in an epidemiological lab. So I'm Richard, and I'm with my colleague Quentin. We are both NGS lab engineers at the Lyon Civil Hospital, and we are part of the GenEpi sequencing platform. So I'm going to quickly introduce you to the GenEpi sequencing platform. So first, GenEpi stands for, in French, Epidemiological, Genomic, and Infectious Diseases. We are a microbiological sequencing platform for public health based in the Infectious Agents Institute in Lyon. As you can see on the slide, the platform is composed of two parts. So you can see in blue the virology parts, composed of biologists, engineers, technicians, biology workers, and also a team of bioinformatics scientists. So you can see the whole workflow that we are working on in blue. And you can also see in green the bacteriology mycology part, which is another team of the GenEpi platform, composed also of biologists, engineers, and technicians. So basically, both teams are sharing the same infrastructure and the same labs. So let's get started with the presentation. So everything started in February 2020 with the Contamine Montjoie first French cluster of SARS-CoV-2. In our lab, those first patients were sequenced using a metagenomic method, which is an untargeted sequencing method. Basically, this is the reference method for sequencing mutations. First, it's a laborious manual pipetting, which induces high contamination likeliness and also a limited throughput, obviously. More important, it has a very limited sensitivity, which means that for samples with a CT value over 20, you can basically not really hope to have a great quality sequence. And so in 2020, our lab could generate around 100 sequences per week, as you can see on the scheme, which is on the right side of the screen, around here. So from February 2020 to around 20, the lab would run at 100 sequences per week. Later on, in 2020, a targeted sequencing method came out, which is basically the COVID-sec assay, which is an amplicant approach. So it shows better sensitivity compared to metagenomic, so the first method that we use in the lab, and it has a high potential for high throughput. So to quickly present you the technique, the Illumina COVID-sec basically consists in cDNA amplification from RNA SARS-CoV-2 using two primer pools. You can see again on the right side of the screen the amplicon approach that was used in October 2020. You can see that the sequences generated from our lab slightly increased at that time, and you can see here the first alpha variant detection in our lab. Speaking of which, in late December 2020, we have detected our first alpha variant, and also the first alpha variant in France. It was detected in a nosocomial cluster of the hospital, And importantly, the alpha variant bears H69V70 deletion spike, inducing a false negative for the S target of a PCR kit. This particular characteristic showed great interest in the sequencing process, and so a targeted sequencing strategy set up on the national scale was made in response of a lack of sequencing capacity at that time. So following this, in January 2021, the Emergent Consortium was created. So basically 4 public sequencing platforms were set up. So at the Institut Pasteur, in our lab in the Hospices de Lyon, APHP, and IHU in Marseille, with Santé Publique France, which is basically a health authority in France. For the 4 public sequencing platforms, there was one main goal, which was the SARS-CoV-2 variants identification and circulation surveillance. I'm going to let Quentin explain you the workflow that was set up for this goal.
So thank you, Richard, for this complete and detailed introduction. Hello, everyone. So a few words about the large-scale sequencing activity evolution. At the beginning, between 2015 and 2020, the NGS platform was composed only of 4 members, including 3 biologists and 1 technician, especially dedicated to research activity. So due to SARS-CoV-2 pandemic, activity was focused to routine diagnostic, leading to team growth with actually 18 members, including engineers, data managers, bioinformaticians, and biologists. So it also led to equipment growth with the arrival of new automatons for the extraction process as the Hamilton started, or the MGI 1960P, and also automatons for all pre and post PCR processes with the acquisition of HSPT automaton as the Mosquito or the Dragonfly, but also the Hidimina Novasec 6000. So this power-up allowed us to develop and design an automatized and miniaturized process with HSPT automatons. And this workflow is split in 3 different parts. So the first one is the extraction process. So individual samples are sending to a lab coming from clinical hospital or private public laboratories in different format of tube. So they will be reformatted into a 96-well plate containing those clinical samples with the addition of positive and negative template control. So the Hamilton will do this process twice, resulting in 192 samples per run. So then those 96-well plate containing clinical samples in control will be transferred to the MGI 9060P in order to extract the SARS-CoV-2 RNA. It will resist 2 different plates, 96-well plate containing our RNA extract from samples and control associated. Then we can skip to the pre-PCR part using HSPT automaton. So we will pick 4 different 96 RNA extract plates coming from the MGI in order to reformat them into 384 wells plates to begin the miniaturized automatized COVID-sec process. So in short, the COVID-sec pre-PCR process is a reverse transcription of the SARS-CoV-2 RNA in order to obtain cDNA, and then we will apply a multiplex PCR with 2 different primer pool, 1 and 2, to result in a PCR amplification of the SARS-CoV-2 genome. So for this step, all the different reagents and PCR mixes will be distributed by the Dragonfly in 384 plates with miniaturized volumes. The Mosquito will be used to do all pipetting steps in miniaturized condition as the RNA transfer to the cDNA plate or the cDNA transfer to all pooled primer plate. Then we can skip to the post-PCR workflow using HSPT automatons. So the first step is to batch the 2 pooled plates that we produce in the pre-PCR pods into 1 library 384 plate. We will have a whole SARS-CoV-2 implicant to this library plate. So in the same idea that the pre-PCR part, reaction mix distribution will be directly into the library 384 plate. We will also use the Dragonfly to distribute all wash solutions needed in the process on new 384 LSBD plate from HSPT. So as the pre-PCR part, the Mosquito high volume will be used to do all pipetting steps, including washing steps that are needed in the process, but also the 384 indexes distribution directly into the library plate. So in short, the COVID-sec post-PCR part is a stage where the DNA will be linked to a B-linked transposome in order to be fragmented. So the DNA will be cut and we will add a sequencing primer that you can see in blue and in green. Then the indexing step will be the addition of 10 nucleotide barcode that you can see index 2, index 1, with the addition of sequences required for the sequencing cluster generation P5 and P7. So this is a sequencing ready fragment that will be used for the sequencing process coming from the post-PCR from COVID-sec. So then we can go on the sequencing process. So basically, the post-PCR workflow will be applied 4 times in order to obtain 4 different 384 libraries, so 1536 samples. So we will pick 768 samples coming from 2 different libraries with 2 different indexes combination, and we will load those samples directly on NovaSeq SP flow cell lane with the XP protocol from Illumina. We will do the same process on the other side with the remaining sample in order to have the 1536 sample run on our NovaSeq. I will let Richard talk about the miniaturization experiment that we did on this workflow. Thank you.
All right, thank you, Quentin. Okay, so for the miniaturization part, we did a simple experiment to compare the results from the automatized and the manual process. So basically, the experiment consisted 20 samples plus 3 negative controls plus 1 positive control, looking like the experiment that we are doing every day, just with lesser samples. So the 2 conditions were applied, so the miniaturized automatized COVID-sec process with the SPT automatons and the full volume manual COVID-sec process. Both libraries were sequenced on the NovaSeq, and the results that we obtained are the following. So first, let's start with the coverage percentage. For those who are not familiar with those aspects, the coverage percentage represents the number of reads aligning to the reference genome sequence. So you can see here on the left side of the screen, the box plot representing the coverage percentage for the 20 samples that I've talked earlier. You can see that in the miniaturized conditions, compared to the manual, the coverage percentage seems to be at least a little bit better than with the manual conditions. And if you see on the right side of the screen with the linear regression, it's just another way of representing those results. But you can see that again, the miniaturized process seems to be a little bit better. And more importantly, with the late CT, as you can see around here, around the 30, you can see that we have better performances with the miniaturized compared to the manual process. To add a little bit to those results, we can also use the depth of the sequencing depth that we were able to get with those conditions. So again, on the left side of the screen, you have the box plot representing the sequencing depth. For those who are not very familiar with that concept, is the depth is the number of unique reads that include a given nucleotide in the reconstructed sequence. So here with the miniaturized, you can see that basically the depth is a little bit better or more homogeneous with the 20 samples. And again, on the right side of the screen with the linear regression, the results are very consistent with the precedent graphs, where the miniaturized seems to have better overall performances on this set of samples. So next, we're going to talk a little bit about cost reduction. Obviously, by miniaturizing the process, we are using less buffer, less reactives, etc. So the initial cost per sample with the manual process was around $20, considering the COVID-sec price of around $11 and the NovaSeq price of $7 per sample, using a 384 samples library. When we automated and miniaturized that process, the price for the price per sample decreased to $15, around between $15 and $16, which represents a 14% cost reduction. The cost reduction applies on the COVID-sec price, because this is the COVID-sec that was miniaturized. And when multiplexing even more with with 1,536 samples. This time, this is the Novasec price that is decreased, leading to a price of $10 per sample, which is a 44.4 cost reduction compared to the manual process. We can also talk about time reduction, obviously. As I said, the manual process is very time consuming. It's a very laborious experiment. And so at the time where we have done the experiment by hand on the manual process, the RNA extraction will take basically 1 day. The pre-PCR step will take 2 days, and the post-PCR sequencing step will take around 4 days. So the full process would take about 1 week for less than 100 samples. By automatization, we could do 384 samples with the RNA extraction around 3 hours with the MJE and the Hamilton Starlet, the pre-PCR and the post-PCR in around like 25 hours combined with the SPT Lab Tech automatons, giving us a full wet lab process of 28 hours for 384 samples. For 1,536 samples, the full lab process will take around 41 hours. So this is a great increase compared to the manual process. So overall, this is the graph that represent all the sequences that our lab have produced since today. So you can see the different steps that we have used. So the first, the metagenomic, with the first contaminant cluster, then the amplicon approach that we have adopted, and then the automation of that amplicon approach and the miniaturization. So we have reached several milestones with the 10,000 sequence, 30,000 sequence, and 100,000 sequences. Finally, in late 2022, we have also detected quite several new variants with the Gamma, Delta, Omicron variants, and overall, the automatized and miniaturized amplicon approach allowed us to have a mean of 2,500 samples per week, and during the highest activity in August 2021, we could do up to 5,000 sequences per week. I just want to add a little bit more of information on the emergency branch, that you might have seen on the GNAP platform presentation. This is a parallel workflow that we are doing for lower throughput. So basically, we do the workflow extraction in 1 hour, the pre-PCR in 4 hours, and the post-PCR, so the library preparation in 1 hour for up to 96 samples. We perform the sequencing steps on the GridION from Nanopore Technologies, and the full wet lab process and the sequencing for up to 12 samples would take less than 12 hours, and up to 96 samples would take less than 20 hours. So this is like the rapid branch of our lab. I'm going to let Quentin finish that presentation, and thanks for the attention.
Thank you, Richard. So, all right. So all these sequencing processes are done in collaboration with so many different health departments, National Health Department, like Santé Publique France or the Agence Régionale de Santé, but also with other HCL clinical services in order to do a complete and efficient variant identification and monitoring with potential impact on SARS-CoV-2 antigenicity, transmissibility, and virulence, and also to use what we call targeted sequencing as a diagnostic tool in order to monitor the effectiveness of treatment, especially monoclonal antibody, as Richard described with the emergency branch, in order to identify vaccinal failure or serious cases of SARS-CoV-2 reinfection that we observed at the hospital. Also to realize some cluster investigation linked to the hospital or to the population. We also use it to monitor the effectiveness of RT-PCR tests that are done in clinical hospital or private or public laboratories, and also to monitor the chronic infection in the immunocompromised patients leading to variant emergency. So to conclude, as a response to SARS-CoV-2 pandemic, we developed and designed an efficient and high-throughput well lab process with miniaturized volumes and significant cost reduction. So it allows us to have the highest sequencing capacity in just platform in France during the epidemic peak, with a maximal theoretical workflow of 10,000 sequences per week. So we can also apply this process to other infectious applications as the monitoring of influenza virus, the RSV HPV viruses, enteroviruses too, and also to do some drug resistance with sequencing for Staphylococcus legionellosis and other bacteria. We also have a publication linked to this work in progress, which is the automation and miniaturization of Illumina SARS-CoV-2 whole genome sequencing protocol by the NGS GNP platform team. So thank you for your attention. Thank you for watching. Thanks.
Okay, thank you, Quentin. Thank you very much, Richard, for this presentation. I will just go very briefly through the technology that was used and what are the other possibilities of our liquid handlers for the NGS laboratories. So SPT Labtech is the company that was established approximately 20 years ago as an engineering company, and we focus on designing and manufacturing innovative solutions for sample management, cryo-EM, but mainly liquid handling. Particularly in genomics, we focus on collaborative nature of working with users of our liquid handlers. We use our engineering background and the scientific background from our users to create the liquid handlers that can dramatically reduce cost per sample without compromising the quality or even improving the quality, enabling the life scientists to generate more data and accelerate research. In terms of the liquid handling solutions that at the moment we offer is a broad range suitable for different applications, for different throughputs, for different needs. So we have the range of the apricot instrumentations, which are pipetters, either 9684 or pipetting by column. They use the classical air displacement to transfer the liquids, and they find their way in NGS labs, either for the simple reformatting of the cells or supporting a little bit more complex solutions. We have also mosquito and dragonfly discovery, already mentioned by Quentin and Richard. These are unique instruments that use true positive displacement technology to deliver low volumes of reagents or samples. And more recently, we launched Firefly, which is a compact platform that combines air displacement pipetting using up to 96 tips with non-contact dispensing, but also it includes shaking and incubation options. Going forward, I will focus on mosquito and dragonfly. So in a traditional way, how the liquid handling world is, there are air displacement. There are multiple approaches, workstations, pipetters, manual pipettes, walkaway solutions. Most of them, they use the air displacement or liquid displacement pipetting. This technology has some limitation, especially at the lower end of pipetting volumes. Where mosquito is, it's filling this gap between the picoliter or nanoliter dispensers and the air displacement pipetting solutions. So its optimal accuracy and reliability is between 25 nanoliters up to 5 microliters, perfectly fitting into this existing gap. What's unique about the mosquito is our tips, is the pipetting. So as mentioned previously, it's a true positive displacement pipetting technology. Tips can be in 2 volume ranges, either between 25 nanoliters up to 1.2 microliter for the, let's say, single cell applications. More commonly in NGS laboratories, the range of the high volume so between 500 nanoliters up to 5 microliters is more suitable. Tips are disposable, gamma irradiated, ensuring that there is no contamination issues. The true positive displacement technology enables transferring the liquids irrespective of the viscosity of the liquid, but also to go lower with the volumes down to 25 nanoliters. Mosquito is a pipetter. It works, you can imagine Mosquito as an automated, low-volume, manual pipetting. So it has ability to multi-aspirate, multi-dispense, to mix. The dead volume, both in the source wells, but also in the tips, is negligible, so you don't need extra reagents in your reservoirs. There is no air in the system, so there is no bubbles, there is no clogging, there is no dripping. And if you look at the end of the tip, you can see it's straight barrel, which means that there is no shrink effect, which makes pipetting very gentle on DNA and enzymes. And finally, those tips are like a small syringes that are packed in a very compact way, and you can have on one spool up to 36,000 tips. Specifically for the molecular biology or NGS laboratories, there is a mosquito HV genomics, which comes with the specific consumables and accessories. So we have here the accessories which support reaction setups. So we have cooling blocks, we have specific magnetic blocks for work in a fluidifiable plates. There's also the spool cover that protects tips from the dust, from the environment. Obviously, there are tips which are gamma irradiated, but also with Mosquito HV Genomics, we have the wizard-based user interface genomic software, which helps with setting up for the first time, but also in future, the new applications. Dragonfly Discovery is a non-contact dispenser. Similarly to the Mosquito, it uses the true positive displacement technology. It can deliver liquid to the wells, either using 3, 6, or 10 independent channels. Again, as a true positive displacement dispenser, there is no liquid classification, there are no liquid classes to set up, there are no compatibility issues, irrespective of what kind of reagents you use. The dispense volume is anything between 200 nanoliters up to 4 milliliters. There is no situations of clogging or blocking of the nozzles, and it's an extremely quick instrument that can deliver liquid to fluidifiable plate in less than 30 seconds. It uses a specific reservoirs with loaded volumes. It works with the different SBS plate formats, 96, 384, 1536, and there is no calibration or maintenance. Irrespective of the volume dispense, it's extremely quick. I already mentioned that. So in this particular video, you can see dispensing from the 6 heads. So this is a 1 microliter drop placement on the flat surface. And all of those syringes are firing at the same time to deliver liquid as quickly as possible in a fluidifiable format. It's also possible to dispense into 96 well plates. And in this particular example, we have 100 microliter dispense into 96 well plate. The number of syringes here used is 3. And as you can see, whether it's a 1 microliter dispense or 100 microliter dispense, in fact, there is not much difference in terms of the delivering the liquid or filling the full plate. Similarly to Mosquito Genomics, there are accessories that work with Dragonfly. So the magnetic block and cooling block earlier mentioned for the Mosquito, they are compatible with the Dragonfly, obviously. Dragonfly Discovery uses reservoirs as the source of the reagents. This can be delivered as a gamma irradiated sterile, similarly to the syringes. For the solutions for the reagents that are sensitive to temperatures, we can also provide the passive cooling for the reservoirs. And finally, for the setups where the higher volumes are required to be dispensed or more in a production environment, instead of using the single consumables like reservoirs, there is possibility to have the pump system. So together with the peristaltic pump and with the tubing, there is an auto-feed system to fill the plates. Quentin and Richard already mentioned how mosquito and dragonfly help them with increasing the throughput in their lab. But in the last 10 years, we work with several different laboratories, and we were able to implement and validate several different methods on our instrumentation. So you have here the small selection of different methods that were already published or shown that are working well at the decreased volumes. This includes several different application areas. We have the single-cell RNA-seq, we have the single-cell DNA-seq, but also going downstream, we have the library preparation for the RNA, we have library preparation for the RNA, and these examples include kits from NEB, from Invitrogen, from Illumina, but also more unusual approaches like amplicon sequencing but also we support the Olink Explore for the proteomics. So I would like to thank you everyone for your time, and we are all happy to answer any questions you may have. Thank you.
Great, thank you, Kamila. As a reminder to our webinar participants, if you have a question, you can type it into the Q&A box in the control panel. We'd like to ask our attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. We'll now start the Q&A. So the first question is for Quentin and Richard. What is the per sample cost for your rapid long read launch?
Yep. Can you hear me?
Yes, I can hear you. Do you need me to repeat that?
Okay, so I must correct the question because I guess the long read mention is because we are using the nanopore sequencing device, which allows long read sequencing. For the rapid branch that we use, it's not very much about long read because the protocol that we use in nanopore use fragmentation as in the COVID-sec, as described in the COVID-sec. But the cost for it obviously it depends on how many samples you pass in one run. I did a quick math on the 96 samples libraries, and using our wet lab process, the total per sample was around 12 euros.
When applying the miniaturization, how did you decide which reagents you can reduce to have robust results?
Basically, we did a lot of trials, determining which reagents we could miniaturize or not, and in the end, we found a combination that was working pretty well, and that gave us the results that we presented, comparing the manual full volume and the automatized miniaturized process. So it was all about trials, modifications, and finding the best combination possible, and that's what we did in the lab.
We have a question here asking for a link to the bioRxiv paper. We might have to follow up with the person asking that to get them the link, but can you give the title of it?
Quentin gave the title of it, but yeah, oops, sorry. Where it is.
While you grab that, we can go to a question for Camila.
This is the title. We can share the link with the person that has the question later, because I could not send it right now.
Okay. Yeah, we can follow up.
We will do a follow up as soon as possible.
For Camila, how easy or difficult is it to program mosquito and dragonfly?
Well, probably it's a better question for Quentin and Richard, but what we strive is to create the software that supports biologists at different levels. So you don't need automation background, you don't need the programming skills to create the protocol on both mosquito and dragonfly. But I will let Quentin and Richard to comment further as they spent some time in front of screens to create those protocols.
Yeah, so as Camilla said, the software is quite friendly. It's quite easy to use, and we did you can basically modify the program right in front of the machine and do everything you want to test different programs and conditions. So as Camilla said, everything is on the computer, and it's quite easy to use, easy to master, and yeah, you can just play with it and find solution that works.
What are the limitations of the wet lab workflow?
Yeah, sorry. The main limitation with our workflow is as the activity grows, like during the highest activity, for example, in August 2021, where we did 5,000 samples in a week. Obviously, it's the contamination, so we need to follow up that contamination very strictly. And as the machines are open, we need to make sure that the contamination are completely under control. So by decontaminating the automatons, by following the global contamination degree, and so this is the main limitation of the process is the contamination.
Got it. How do you think you can improve your throughput even more with your wet lab process?
To improve it, what Quentin presented to you was the limitation that we have in the lab today, right now. So with the resources that we have today, we could enhance again that process by, for example, with as you can see, the UDE unique dual indexes here. What we could do really easily is just order new ones, like set 1, 2, maybe set 3 and 4, and we could multiplex even more libraries in one flow cell. If that flow cell, the SP flow cell, become insufficient, we can order the bigger ones from Illumina, like S2, S1, or even S4, sky is the limit. And this is a process that we could really easily enhance a lot on the theoretical aspect. That's why Quentin said that we could do theoretically around here. I don't really there. Yeah, there. That's what Quentin said. We could do theoretically 10,000 sequences a week for now, by doing 3 times 1,500 libraries, and we can do that on both lanes of the Nova, because the Nova has two lanes, so you can launch sequencing experiments 2 times. So basically, our theoretical limit today is 10,000 sequences a week, but we could just break that limit really easily and go way above that.
We have a little more time for questions here, so if anyone else has questions, you can go ahead and type them into the Q&A box. For Camilla, Richard was talking about the contamination risk. Can you talk a bit about how the equipment handles contamination risk?
That's a very good question, and quite often is asked, especially by the biologists. Contamination can go undetected, so definitely it's something that we need to think before loading the samples on any automation solution. So there are a couple of the things that we focus when we already were designing the instrumentation. So in case of the dragonfly discovery, one of the priorities was obviously speed and delivering the liquid to the wells, but also ensuring that there's absolutely no splashes. And with the correct settings, with the correct height between dispensing and the liquid, we make it possible. In contrast, for the mosquito, whenever there is any liquid left in the tips, it's sucked back before the tips are moving, which massively reduces any possibility of the contamination. And obviously, providing the sterile consumables further decrease any kind of chances of contamination or cross-contamination.
Oh, excuse me. It looks like that's all the questions we have for now. If any of our viewers have any further questions, feel free to send them in and we can try to follow up with our experts. We'd like to thank Richard Chalvignac, Quentin Semanas, Kamila Koprowska, and our sponsor, SPT Lab Tech. Actually, we've got one more question here. Why don't we? Oh, two more. Okay, now they're coming in. Why don't we get to these and then we can wrap up. This one I think is probably for Quentin and Richard. Do you worry about optical contamination when adding more samples to the sequencing runs? Is there an upper limit for number of samples to include on one run? And then Kamila, if you have anything to add to their answer as well.
Currently, we did not reach the limitation. Sorry.
We can't quite hear you.
Sorry.
Yeah, no problem.
Can you hear me?
Yes. Perfect.
Okay. So maybe can you repeat the question, or just I'm going to read it again?
Sure. Do you worry about optical contamination when adding more samples to the sequencing runs? Is there an upper limit for the number of samples to include in one run?
Okay. So for now, we do not worry about the optical contamination, but the NovaSeq is able to deal with really a lot more samples that we are currently using. After that, it depends on the flow cell that you use. As I said, we could upgrade the flow cell or the flow cell with the indexes that we use for the routine. But for now, we didn't had any issues with like saturating the Nova with too much samples. For now, it did not happen in the lab. Maybe if you go too high with and you ask too much for maybe like the SP flow cell, you could have issues like that. But for now, this is not something that we have encountered.
And what is the read depth per sample at the current multiplex using the XP flow cell? And what is the variation per sample?
Okay, so again with the Nova, like to be perfectly honest, we are kind of underusing it. Like the Nova is able to get a lot more samples in one run. So the depth per samples is not something that we worry about. As you can see in the result that we had, we have way enough data and depths for all of our samples using the NovaSeq. So maybe this is something that we would encounter if we put a lot more samples in our runs. But for now, this is not an issue that we have encountered.
Got it. Okay, so that if anybody else has any additional questions, we can try to get our experts here to follow up with you. We'd like to thank Richard Chalvignac, Quentin Semanas, and Kamila Koprowska, and our sponsor, SPT Lab Tech. If we didn't have time to get to your question, we'll follow up. As a reminder, please look out for the survey after you log out to provide your feedback. If you miss any part of the webinar, or if you'd like to listen to it again, an archived version will be emailed to all attendees. Thank you very much for joining us.
Thank you. you.